The objective of Sylvestre Bonnet, a chemist, and his research team is to develop a chemotherapy treatment that can effectively combat cancer cells without causing harm to the body. Chemist Xuequan Zhou made progress towards achieving that objective during his doctoral research. The researcher synthesized molecules that possess the ability to spontaneously form nanoparticles after being introduced into the circulatory system, ultimately leading to their accumulation in the neoplastic tissue. The tumor is subjected to focused irradiation utilizing visible light, resulting in its destruction. The findings of the study have been formally disseminated in the peer-reviewed academic journal, Nature Chemistry. According to Bonnet, traditional cancer medications frequently lack specificity in distinguishing between healthy and malignant cells. Both of them are killed. The investigators have devised a resolution to this issue by utilizing nanoparticles that selectively home in on the tumor and exhibit activity solely in response to visible light. The application of anti-cancer phototherapy enables medical practitioners to target a particular region of the body while minimizing harm to the surrounding tissues. The technology has already been implemented in multiple healthcare facilities. Molecules that have the ability to self-assemble into nanoparticles. Previously, chemists were required to initially affix the chemotherapy agents onto nanoparticles within a laboratory setting. Subsequently, medical professionals administered the aforementioned substances through intravenous injection into the patient's circulatory system. The process of conjugating nanoparticles facilitated the localization of chemotherapy agents to the tumor site. The drug developed by Zhou exhibits a distinct mechanism of action. According to his statement, the laboratory work is deemed unnecessary. The molecules can be administered directly. Upon entering the bloodstream, nanoparticles undergo spontaneous self-assembly. According to Zhou, there are multiple benefits associated with this. Initially, it reduces a substantial amount of labor and planning duration. Moreover, it is also deemed to be a safer and more efficacious approach. The process of synthesizing nanoparticles within a laboratory setting is a complex undertaking, as it invariably results in the production of a heterogeneous mixture of particles characterized by varying sizes and, consequently, distinct physical and chemical attributes. Accurately ascertaining the exact composition of said mixture presents a challenge. It is uncertain to what extent these particles will behave within the human body. According to Zhou, the process of determining the purity of a molecule is relatively simple, as chemical analysis can be employed to ascertain its purity. According to Bonnet, the injection of said molecules into the bloodstream yields nanoparticles that exhibit a high degree of similarity. This is due to the fact that the molecules are processed uniformly by the human body. The molecule synthesized by Xuequan is classified as a palladium complex, which is characterized by a metallic core composed of palladium. Typically, the palladium atom exhibits a tetradentate coordination with 4 nitrogen atoms. However, Zhou's research involved the substitution of 2 nitrogen atoms with carbon atoms. Upon exposure to green light, the palladium complex undergoes excitation and acquires additional energy. The surplus energy prompts the complex to facilitate the transfer of electrons to the pre-existing oxygen molecules, O2, within the irradiated cells. This mechanism generates a type of highly reactive molecule known as reactive oxygen species, which induces apoptosis in cancer cells. Zhou developed a self-assembling cancer drug in the year 2020. The speaker asserts that the newly discovered molecule represents a significant advancement. The activation of the drug can now be achieved under green light instead of blue as a result of the binding of 2 carbon atoms to the metal. The utilization of green light in therapy for mice is more advantageous due to its enhanced ability to penetrate body tissue. Bonnet stated that the ultimate objective is to develop a pharmaceutical agent that exhibits efficacy under infrared radiation. The illumination in question would facilitate a more profound level of penetration. This would enable the combat of larger tumors situated in deeper regions of the human body. This novel investigation adopted a superior methodology that is more pertinent to clinical practice. In the initial investigation, Zhou et al. Administered the medication intratumorally. According to the speaker, the study progressed by examining the effects of the drug in mouse models through intravenous injection. This approach is consistent with the standard practice in hospitals. The inquiry pertained to the viability of drug nanoparticles under physiological conditions within the human body. Fortunately, such was the circumstance. The molecule developed by Zhou demonstrated a high level of efficacy. According to Bonnet, the tumor receives a 10% dosage of the administered drug. 10% of the administered molecules reach their destination. The proportion of nanomedicines exhibiting such characteristics is considerably diminished. A study conducted in the recent past revealed that the mean value is merely 0.7%. What is the precise mechanism by which this can be achieved? Is it possible for molecules to self-assemble into nanoparticles? Bonnet acknowledges that the information in question is not currently known. It is widely acknowledged that palladium holds significant importance, and Xuequan's research has indicated the potential involvement of blood proteins in this regard. In the absence of said proteins, the nanoparticles exhibit uncontrolled growth, leading to their precipitation from the solution due to their increased size. Therefore, it appears that the proteins restrict the expansion of the nanoparticles. However, the exact mechanism remains unclear. The efficacy of the subject matter is established. This is deemed as the utmost significant aspect. What is the reason for its high level of effectiveness? This assertion necessitates additional research to validate.